Peptide oligonucleotide conjugates for use as nanoscale building blocks. Imagine being able to make nanoscale structures as easily as we build with Lego. This experiment aimed to use two self-assembly principles to make the building blocks needed for a hollow nanobox, like this Lego figure. This could be used to transport drugs in the future. It makes peptide design as easy as building with Lego bricks. Peptide drugs are usually administered by injections due to their poor physicochemical properties, which include denaturation, poor membrane permeability, and large molecular size. This is an inconvenient method for the patient, and the bioavailability is extremely low, often as low as 1%. Nanoscale objects have a wide range of use in medicine. Through sophisticated design, a nanobox could be created which could carry drugs inside it to the exact place it was needed in the human body. This ensures a very high local concentration and hugely improves safety margins. The possible applications of designed nanoassemblies are numerous. DNA origami, the manipulation of DNA strands, allows for complete control over nanostructures. It's possible to make smileys and even a box with a lid that opens and closes. This experiment uses two self-assembly principles to make something called peptide oligonucleotide conjugates, or POCs. The POCs are the building blocks for larger nanoscale structures. Almost like Lego bricks, they enable chemists to build and design new structures. The two self-assembly principles are the fact that the two parts of the POCs, here depicted in green and red, are able to stabilize themselves and each other to create higher-ordered protein-like structures. The first thing to do was to synthesize the acetopeptide by solid-phase peptide synthesis. This is an extremely efficient method to create designed peptides. The next thing was attaching the protection group BTN to the oligonucleotide. Then the acetopeptide and the oligonucleotide were coupled together in a click reaction, furthered by microwave irritation. This gave the peptide oligonucleotide conjugate POC. The POCs are created easily and effectively. Once we have the peptide oligonucleotide conjugates, the three POCs can hybridize to form a very stable nano building block, which is stabilized both through the coil of the peptides, here shown in green, and the triplet assembly of the blue and the red. These double stabilizations are the two self-assembly principles we employ. By mixing several strands of these building blocks, their designed abilities will form a hollow nanostructure. This hollow nanobox will be able to transport drugs inside it in the future. The POCs and the nanobox were characterized with small angle X-ray scattering, a method that yields information about the size, shape, and internal structure of disordered and partially ordered systems. The POCs were created successfully as nanoscale building blocks. The SAC studies showed how the POCs formed dimeres and trimeres successfully. They also showed that the POCs were formed into the nanoscale box, showing how the use of two self-assembly principles could be a valid route for optimizing drug delivery in the future. This could open new avenues for circumventing the poor physicochemical properties of peptide drugs. By avoiding the digestive system, this method will enable even drugs with very poor physicochemical properties to be used as medicines. It shows that oligonucleotide helix formation and the coil of peptides can be used to direct nanostructures. The two self-assembly principles work. Both the POC building blocks and the nanobox was created as shown by the SAC's measurements. The highly stable coil and triple helix makes peptide synthesis almost as easy as playing with Lego bricks. This could be used in the future to transport drugs, which would improve safety margins by ensuring high local concentrations and circumvent the poor physicochemical properties of peptide drugs. In short, this could open new avenues for the future of drug delivery.